acquire new skills or insight or broaden your perspective. My background, as far as these two concepts, I'm a subject matter expert in sterilization, and sterilization processes always require validation. I've also written numerous protocols and audited numerous validations for diverse processes. I've also developed responses to inadequate validation findings from audits and failure investigations, and I've helped to author some of the related guidelines and procedures that are scrutinized by FDA and the ISO auditors. The purpose of this talk is to clear up some misconceptions that are prevalent in the industry and enable those using these elements to really add value to their organizations by assuring both products and processes are more robust. If the verification and validation are done right, the process and product should always conform unless there is an obvious equipment malfunction or human error. If design verification and validation are done right and only conforming product is released, then the customer should be completely satisfied and sales will be predictable. From management's point of view, that's not too much to expect for the time and investment dollars allocated to these activities. So our agenda for today is, there we go. I'll let you read it. Today we're going to discuss a little bit about the history and differences between these two items, or these two terms, as well as how to use them to our advantage to prevent future problems. You may also see an alligator pop up from time to time. This is a reminder that the alligator egg you lay or hatch today may eventually grow into that huge 15-foot alligator that will eat up your profits tomorrow. If you want to learn more about alligators, we have a very nice recording on root cause analysis available through compliance training, and you can check that out if you want to see more. But first, we need to understand how we got here, and beginning with what some call the age of ignorance. Okay? Until the 1980s, sterilization was monitored using the U.S. pharmacopoeial sterility test, where 20 units were tested after sterilization, and the lot was rejected if one or more were positive for growth. If there were no positives, then sterility of the lot was verified. Well, let's consider some basic sampling statistics. This table defines the typical operating characteristic curve showing the probability of rejection based on the sample size and the percent of the lot that's contaminated. If, for instance, the lot is X% contaminated across the top, and you take N samples down the left over here, then the number in between is the probability that you'll catch one. Okay? This second line here, whoops, I got to click on the right thing. There we go. The second line here, that's the criteria for the USP sterility test, where a sample size equals 20 and you accept on 0. Notice that the lot would have to be over 3% contaminated before the test would have even a 50% chance of catching it. To put this in perspective, a 0.1 contamination level is equal to one non-sterile unit in 1,000. But the sterility assurance level for sterile products is less than 0.0001%, or less than 1 in a million. To detect 1 in a million at a 90% confidence, we need a sample size of over 100,000 units. That's just impractical. Therefore, with any reasonable sampling plan, we can only detect gross contamination, and the test can't be used with any confidence to verify the required SAL. But the test gives a warm, fuzzy feeling to management and the FDA, and believe it or not, final testing is still required for many drug products. The conclusion is that post-sterile testing has no practical quality assurance value for monitoring sterilization. It only detects gross process failures, again greater than 3% contaminated half the time. And the problem with most testing programs is you're never sure that you tested the worst case. It tells you you can sterilize something, not that you did, or if they are all sterile. This is why today post-testing is only used during validation or audits. There's my thing. There we go. All right. It's hard to imagine, but validation was not widely used prior to the 1970s. The prevailing thought at the time was that everything needed to be inspected to assure conformance, and as long as it passed the inspection, it was okay. Today, through process failures, complaints, and recalls, we know this to be false. We cannot inspect quality into the product. So the FDA began to require validation for any process that could not be easily verified, like sterilization, And it took some time for people to really understand this, and even longer to convince people that it could add value. If the verification and validation are done right, the process and product should always conform unless there's an obvious equipment malfunction or human error. If the design verification and validation are done right, then, as I said before, the customer will be completely satisfied and projected sales growth will be achieved. And again, for management, that's not too much to expect from putting all this money into a project. Now, what I'd like to do, and I don't know if it's possible to do it this way, is it possible, Alan, if it's possible for them to raise their hands, or maybe even just put it in the chat box, just tell me if